Hello and welcome to the Federal. We have with us TV Venkateswaran, Senior Scientist at Vigyan Prasar, a Government of India initiative to popularize science among the masses. Venkateswaran is a familiar face both on national and regional television channels, and he's rather busy nowadays, crisscrossing the country and explaining issues related to coronavirus pandemic, which has unfortunately seized all our lives. He's one of the finest science communicators the Indian scientific community has produced. He can break down complex issues and explain them in an easy, understandable manner. Welcome, Venkateswaran.
Thank you. Thank you.
Let's first talk about coronavirus, its mutation, and testing. RT-PCR test is considered as a gold standard as far as identifying the virus is concerned. Through this test, we know whether a person has been hit by the virus and if so, to what extent. There could be margin of errors, like all other tests. But the test so far has proved to the most reliable way of identifying the disease. But with virus mutating into, various new forms, it's being said that it can even escape the test. What are the reasons?
Okay. Let's try to understand what do we mean by when we say that RT-PCR test in a kind of a very simplistic fashion. I mean, very simple fashion. Let's say, for example, for the purpose of understanding, we are in a library, and unfortunately, for some random reason, there is no book cover. The books don't have any cover, so we don't know the title of the book. Okay. So we want to find what this book is about. So we flip open a book, okay, and I read there, for example, one couplet from Tirukkural, Agara Mugara Ezhuthallam, Adi Bhagavan Mudatre Ulaghu. Okay. Then I come to a conclusion that, oh, this should be the book on Tirukkural. Yeah. I don't read from the first page to the last page.
Right.
Similarly, for example, the novel coronavirus has a genome called RNA, which has about 30,000 alphabets, the genetic alphabets. I mean, if it is a DNA, we will say that ATGC, but if it is RNA, I mean, you have U, so instead of T. So there is 30,000 alphabets. You can't read all the 30,000 alphabets and check whether it matches with your reference, because then it's a very time-consuming process. It's a costly process. So what you do is you pick out some portions of that 30,000 alphabet and then check. For example, recently, about a few days ago, I did a RT-PCR test. And in my RT-PCR test, which was actually, for example, designed by the IIT Delhi, called COVID Sure, what they are doing is that they have taken snippets from ORF1A, ORF1B, N and N2G, okay, of the novel coronavirus. And those parts, they will check whether the sample that was taken from me have those parts. So if they have those parts, then you conclude that you are positive. If none of those parts are available, then you conclude that it is negative. Yeah, now you are talking about the variation. There could be a possibility that, for example, because of some printing error in some book, one couplet of Tirukkural might be wrong. But when I'm going to pick out some 4-5 couplets, then it's not that, for example, all the 4-5 couplets will have mistakes, right? I mean, for that to happen, it's going to take a lot of time. So, for example, some of the tests which are available use primers, which actually picks up different parts of the genome. But some of the primers pick only, for example, the spike protein. So those RT-PCR tests may not give you good results. One recent study kind of pointed out that close to about 2% of RT-PCR studies might give false negatives, meaning that you might have a disease, but the result may show negative. That is why nowadays, for example, if you read the results very carefully, they also write that although this is negative, if you have symptoms, consult your doctors, and clinically we need to also look at it. That's something that is done. You can change. You also say that can you change the RT-PCR test? You can change. You can change the primers. Problem would be that you might have produced large amounts of primers earlier. It may be in your stock. maybe you don't want to waste them, and also producing new would also be time consuming. So at some point of time, maybe you might say that, I mean, this kind of error I am willing to live with. I mean, it's a kind of a decision that needs to be taken based on this kind of multiple factors.
So what do you do when a new kind of mutant comes into the atmosphere, or we learn that that's a new kind of virus which has come? So do you recalibrate, or what do you do with it?
Yeah, for example, some of the tests which are available in India, like for example, the one that is developed by IIT Delhi, and some of them can be recalibrated. I mean, they can be very simply recalibrated, but some of the tests cannot be recalibrated that easily. That requires a lot of change.
Now, how
Uh,
quickly it reaches from lab to, you know, from
Yes, the kind
testing
of
centers?
technology, the kind of primers that you are using, the kind of kits that you are using makes a difference. For example, with the same person, if you use different kits, sometimes, for example, one kit may say negative, but the other kit may say positive, if the person is actually positive.
Okay.
I mean, in India, we don't use one kit. I mean, we use multiple kits because we are a very huge country, and we had imported quite a lot of kits from different parts of the world.
So it's a challenge actually. Managing all this is a challenge.
Yes.
How are we dealing with this? Because India is such a vast country. If one change happens in one part of India, and that has to be communicated, how does this happen?
Yeah. So actually, if you look at the Indian scientific establishment, they had with foresight expected this thing to happen, because any scientist worth a salt would know that the virus would evolve. I mean, it's not like one virus and that's the end of it. We know that it will evolve. I mean, even early in pandemic, we knew that every 15 days there is one mutation in the virus, roughly. That's the order of change that happens. Therefore, in India, they had established what is called as digital monitoring, basically, and genome monitoring. Basically, they selected samples from different parts of the country. They were sequencing those samples and then checking those samples with the primers which are used in India, that is the kits which are using India, to see how much actually we are missing. So basically, the issue is like this. I mean, if you go to a vegetable vendor, the vegetable vendor has a scale, right? The kind of accuracy that you are looking at vegetable vendor is not like, say, for example, gram level accuracy, right? I mean, even if about 5 grams here and there, you don't mind. Even 10 grams, perhaps you would keep your eyes shut. But if you are going to a gold shop and you are buying gold, I mean, even fraction of gram is going to be a big issue, right? So when you're talking about large scale testing in a situation like this, where you are trying to find out who are all affected, so that perhaps they can be given counseling with regard to isolation and so on, uh, there are some margin of error you are willing to live with, because if you are not going to have that kind of margin of error, even you want a very, very well working testing kit, then your cost is going to go up. So you have to balance the cost and the some false negatives that you may have to live with. So at this point of time, what we know is that around 2% of RT-PCR results may show negative, but they might have actually infection because of the changes that have happened in the last about 1.5 years in the novel coronavirus genome.
Right. So when you said this genome sequencing, you need samples for that, and those samples need to come from all over.
Yeah.
Are you getting the samples real time? Are you able to send them back with the testing, the results? How fast is this? Is this process
Yeah.
been speeded up?
So if you look at, for example, this genome sequencing project, this genome sequencing project started in India really very way back. In fact, the very first identified corona case from Kerala, their genome was also sequenced by National Institute of Virology. And subsequently, we kind of embarked on a kind of a systematic project to collect samples from different states, and then today, I mean, we have crossed more than 4,000 samples. Okay, so it's being done.
So
But I won't
it's a
say
regular
that these
process?
samples are coming evenly from all states.
It's a regular process. It happens.
It's a regular process. It's a very regular process. I mean, there is a systematic effort to collect samples from different parts and then sequence. In fact, about the recent results that we see, no, right? For example, a new mutant has arrived in Maharashtra, or a very new mutant is seen in West Bengal, or the UK variant is seeming to be a bit strong in Punjab and Delhi. These kinds of results actually come from this kind of sequencing.
Right. So again, there are two kinds of variants. I'm told a variant of interest and variant of concern. When these things are mutated, how concerning are these varieties?
Yeah.
How many of them? Because there are many variants, about 700. I'm told.
Actually, if you look at lineages, I mean, there are many lineages, but if you talk about every mutation as a change, then there would be thousands of them.
Right.
Because every 15 days there is a small genome change that happens in virus. Okay, so that would be constantly happening, but most of those changes won't be carried forward to the next generation. Mostly what will happen is that within 1 or 2 generations that will die, because the person would be transmitting it to another person. That person would not be able to transmit to another person because that person was in quarantine or some way keeping away from others, is not transmitting because of our human intervention. Okay, social measures like a mask or things like that. Then that variant, that mutation will not survive. Okay, so what we call as variants are when you have a mutation which causes certain significant changes in the virus behavior, let's say so. As human beings, we are interested in three kinds of behavior. There may be many others, but we are not worried about it. We are worried about three. One is that, is it affecting the spread?
Okay.
Whether this change is affecting, whether it is going to increase the spread or reduce the spread. Okay, that is number one. The second is it going to be more infectious? Okay, and the third is whether it is going to be immune escape. These are the three important characteristics one looks for in a variant. When I say when you look for, what we do is that when we sequence the genome, we know what is the amino acid change that has happened. So from the change in the amino acid, we can guess, or rather we can compute theoretically what would be the behavioral change in this variant. So when you see either of these three, that it is going to aid the spread or abate the infectivity or going to escape the immune, then we call it as a variant of interest. But then what may happen is that maybe in that area where this variant has emerged, the people are behaving with consciousness. There is a systematic administration. Then what would happen is that this variant, even if it is having advantage, evolutionary advantage, would not spread. But
So
at times,
how are we? I mean, should we really be get concerned, because a whole lot of them have now becoming more concerning
yeah.
form of.
So we can take two examples. For example, if you remember back somewhere around April, October, that period of last year, we were all talking about this new variant that has emerged in India called AI3.
Right.
Yeah. But because of the measures that was taken during that time, both by the administration and the people, now you don't see that variant at all. That's vanished. It essentially meant that in those places, the measures were substantial. That is, the measure to contain the virus spread was substantial. So we don't see them. But on the other hand, today, for example, you see that the UK variant is spreading in different parts of North India, particularly Punjab and nearby area. So we can very clearly see that the channels of virus spread is still open in that area. So when you both are virus of variants of interest, but AI3 no longer is spreading, so it is no longer concern. But the UK variant is spreading. It's also seen in various parts of the world. So it becomes a variant of concern. So it is a variant of interest only if it shows potential ill effects. It becomes a variant of concern when you actually see in your genome sequencing that it is spreading, it is gaining ground, or it is spreading to new area. Then you classify it as a variant of concern.
Right. You see, one theory believes, that when mutations happen, the virus actually loses its potency. Say, for instance, we are all living with one or the other kind of other forms of infections, say even common cold or flu. But in the case of coronavirus, the varieties, mutant varieties, are only getting stronger. Why?
Yeah, okay. Basically, because we are looking at only the variants which are becoming stronger. We are not right now focusing on variants which are becoming less virulent. That's all. So it is our selective gaze. We are looking at variants which are becoming virulent because right now we are in a pandemic situation and a large number of people are getting affected. They are becoming sick. They are hospitalized. Many of them are dying. Okay. At some point of time, for example, this pandemic will end. Maybe through a large scale immunization, that is by administering vaccines, hopefully. That's what I wish so. Or by its a natural spread. If it is going to be natural spread, then the cost is going to be very huge. Human cost is going to be very huge. Large number of people are going to be affected and a large number of people are going to die. But somewhere, that's the only two end. I mean, there is no other alternative. One of these two things will happen. When that happens, what will happen is that the less virulent novel coronavirus would survive in the background as a less virulent one, which will be appearing once in a while here and there, causing some kind of a cold or some of those kind of symptoms, respiratory problems. That's it. It will become
Both.
history.
Yeah. So you mentioned about vaccination. So in what way that can protect us from mutants? Because even vaccine might have been produced keeping in mind a certain protein,
Yeah.
but what if that itself has changed? It's taken a new form. How does vaccination work? In other words, I want to know if you can explain what is this vaccine escape all about.
Okay, so I'll maybe use a very simple analogy to explain certain things. Okay, I mean,
Right.
when we are talking very popular way, I mean, some of the nuances we will miss out, but that's okay. But for a larger understanding, we can think like
Right.
this.
We are talking for a larger audience.
Yeah, yeah. Suppose you are a car mechanic and you are used to repairing, let's say, once upon a time in India, ambassador cars or Fiat cars. So you are a car mechanic, so you will have a range of spanners with you to open the engine, right? But all of a sudden, there is a liberalization and you have BMWs running all around. So for the first time in your shop, you see a BMW coming in. Okay. So you look at your spanners. Some of them fit the bolts and nuts, but many of them don't fit. So what do you do? You take your spanner, and if it is very urgent, maybe you will tie some twine around it, try to make it fit, right? You will try to
Yeah.
open it. Yeah. But in the meanwhile, you will also call your boy and say that run to the hardware store, get me this particular spanner, this
Right.
specification spanner. So when the particular specification spanner comes, then it's no longer, you can immediately open the engine, right? So basically, you can think of the engine of a car as, let's say, the spike protein of the novel coronavirus. On top of the spike protein, there is something called as antigens. Okay, like for example, the bolts on number of bolts on the car engine. Each of them not same. Each of them having different shape, different size, right? Even if the shape is same, the size is different. Okay, so there are many antigens. So you can think of your spanners as your antibodies. So when we are talking about a variant in an engine, instead of a particular bolt, you have now a new bolt, but the remaining bolts are same.
Mm. Right.
So if I am a car mechanic, I may not have a spanner for a particular bolt, but for the remaining bolt, I have the spanner.
Yeah.
right?
But if
So,
you have a flat, you'll have to remove all the 4 bolts
yes.
or the 6, whatever you have.
Yeah, but point is that you are not helpless. So what happens is that when you are targeting the spike protein, it generates a range of antibodies in you, not just only one. Okay. So because it generates a range of antibody, the vaccine gives you protection, because even if the antibody is not able to attach to one particular antigen, the remaining antigens are neutralized. So the virus is neutralized. So we are cool. Okay. And most importantly, we are looking at what is called as a T cell response when you are talking about a vaccine
Right.
protection. So what is this T cell? I'll just explain in a very simple way. Like suppose when the virus gets into a particular cell, human cell, then virus antigen will be visible on the surface of that cell.
Right.
So the T cells will identify that virus antigen and then destroy that cell. So which would mean that the virus inside cannot make copies, cannot spread to next nearby cells. So now this virus cell, I mean the cell which is infected by virus, is going to have multiple antigen on its body. And the T cell is not looking for only one antigen, any of the antigen. Okay, so if any of those antigens are there, that's enough, it's going to kill it. Okay, so even when variants come, most likely your vaccines are going to be useful. We say that vaccines are going to become ineffective if there is going to be a radical change in the structure of virus. Okay, but in a single segment virus like novel coronavirus, that's very rare. Single segment means, I mean, it's like only one string of RNA. But for example, if you're looking at flu, it has 7 strings. Okay, so you can imagine it like, for example, the novel coronavirus has one single sheet, but whereas the flu has 7 sheets. So when you have 7 sheets, by mistake you can put the bottommost sheet on the top. Okay, so that kind of shuffles can change the flu. That is why, for example, flu, it's very difficult to make a vaccine. And in a lifetime, for example, one person might be infected with 200 types of flu because of this shuffles.
Right. So
Okay.
what you are saying is actually uh, vaccination can save, and it's the best form of protection, the
Yes.
best form of defense one can actually create for oneself,
Yes.
because it can
And all the
actually.
vaccines that we have today are proved to be effective in many ways. Number one, for example, in studies, the clinical trials 1, 2, 3, we know that it protects you from hospitalization and death close to 100%. In fact, very few days ago, I mean, the results from US large scale survey. I mean, this is what is called as clinical stage 4, which usually takes place after the rollout. So actually, in real life situation, when you are giving it to a large number of people, so in US they surveyed 77 million people who have been administered vaccine. What they found is that out of that, only about 5,800 people were infected with the infection, and then only about 77 people died. So 77 out of 77 million. So you can look at the kind of
Yeah,
protection that it gives. So
right.
you can say that it prevents you from death in terms of 99.9999%,
Yeah.
right?
That's very encouraging. Now, I just want you to take to an issue which is concerning all of us, especially capital Delhi, where people are dying because of lack of oxygen in hospitals. It really made me wonder. I read one of your pieces and it came to my mind. At a time when Moxie in Perseverance mission is looking for
Yes.
oxygen molecule on Mars, here back home, Earthlings are struggling for oxygen.
Yes.
How do you see that irony as a scientist?
It's really sad because the technology for producing oxygen has been around from 1905. I mean, if you are looking at, for example, the traditional technology of liquefying air and then producing oxygen, it's there from 1905. And then if you're talking about the modern method of concentrating oxygen from atmospheric air. It's been around from 1970. So the technology is around with us. And in fact, in India, we are producing, if you're looking at oxygen as oxygen, we are producing huge quantities of oxygen. But most of them are not medical grade, they are all industrial grade. But we can tweak these industries to produce medical grade. Of course, then their production capacities will come down because medical grade is a bit stringent and tougher. The production capacities will come down, but you can produce. So, like states like Kerala, for example, or Tamil Nadu are self-sufficient, meaning that not only they are sufficient for today, but the kind of numbers that they have, the production capacities that they have, I'm sure the current crisis they are going to sail through sufficiently well. It's largely to do with many issues, like, for example, mismatch. Where the huge cases have come, we don't have oxygen plants. The oxygen plants are in different places. The medical oxygen production is not distributed around the country in uniform way. Okay, and it's also about 80% of medical oxygen production is done by one single company. These are all some of the issues which I think perhaps we need to relook.
Yeah,
Because,
the administrators will look into it. But as a scientist, I want to ask you, see, we all know that oxygen is very, very important for us. We can't survive without it. But can you just quickly explain, because it concerns the lungs, oxygen is required, how it is processed? Can you just go into the science of it briefly so that we know what happens?
How do we produce the artificial oxygen? That's what you are asking, right?
Yeah.
Okay. Largely, we use two methods. Okay. The first method is called as cryogenic air separation. Okay,
no, that's like one portion of it. You can explain me that. Also, how does a body receive oxygen? What happens to our lungs?
Yeah.
And why does suddenly we become breathless? If you can explain that also. These two issues.
Yeah, sure. So if you look at, for example, we largely use two methods for producing medical grade oxygen today. One is called as air separation method. So it's something like this, we all know that all elements have their boiling point. So suppose if I liquefy the air, so now I have a liquid air, okay? So then I allow it to rise temperature. So when the temperature is rising, at some point, one element in it is going to have the boiling point. So only that will bubble. Like say, for example, if you cool air to about minus 191 degree, the air will become liquid. And then if I allow the temperature to rise to 193 degree, then the nitrogen will bubble and nitrogen will come out. So the nitrogen will come out as vapor, and I can capture the vapor, and then I'll get the nitrogen. So then if I allow the temperature to come up to minus 183 degrees, then oxygen will come out. Okay. And even if I raise the temperature to 0, water vapor will not come out. It still be ice. Water vapor in atmosphere will be still ice. Okay. So it's like a fractional distillation. So by rising temperature, you can take it out. So this is one method. The second method is there are some materials, like say, for example, zeolite, on which if you put air at high pressure, the material will act like a sponge for nitrogen, and it will absorb nitrogen. Similarly, you can have few more material and then remove other parts of components of air, and then you can get oxygen. So the second method is pressure swing method. The pressure swing method is actually a very recent one. I mean, mostly large amount of medical grade new technology is to use the pressure gate method, and that can be also miniaturized, so it can be like your home. I mean, nowadays if you go to Google, I mean, people are selling oxygen aggregators. Basically, it works like that. So this is the way, I mean, you produce the artificial oxygen. The way, for example, our lung works is a very interesting stuff. I mean, when we take breath, we take about 5 liters to 8 liters of air inside. And 20% of that is oxygen.
5 to 8 liters in what period of time?
In
One gun.
yeah.
Oh,
Yeah. So then what happens is that, I mean, that's our lung capacity. Yeah. So then what happens is that 20% of that is oxygen, but not all oxygen is absorbed by our body. Most people think that when you exhale, people think that there is no oxygen in it. That's false. So out of the 20%, only 5% is absorbed by the body. The remaining 50% actually comes out, because the process of absorption is what one can say as a kind of a sluggish process. So what is that process? Is basically in your lungs, there are air sacs, I mean small, minute air sacs. So these air sacs, you have capillary blood vessels, okay, very thin capillary blood vessels, and the blood contains hemoglobin, and the hemoglobin can attract oxygen, something like a magnet can attract a pin, it can attract oxygen. So when the air goes into the air sacs, some amount of oxygen is attracted by this hemoglobin, so the blood becomes rich in oxygen, okay. And because of the partial pressure, the carbon dioxide in the blood vessels come out into the air sacs, and then when we exhale, carbon dioxide comes
yeah.
out. So this is what happens. So if you look at the total surface area in which this whole process is happening, it's 100 square meters, and that's because basically it's called fractal structures, okay. I mean, it's a very interesting stuff. So our lungs are in a kind of a fractal structure.
100 square meter. If you can give me an idea, 100 square meters, how big? Equivalent of
Well,
what?
100 square meter would be 10 meter into 10 meter. So 10 meter would be something like, let's say a I mean, not football stadium. I can't
Football
immediately
stadium would
think
be
of
less than that.
another.
Yeah, okay, fine, I get it. Yeah,
Yeah.
it's
So
really
basically, it's
large.
a huge one because of the fractal structure. Okay, so that's why the surface area is big. It's because of the surface area, the oxygen is able to interact with the blood and then it's able to absorb. So now, when you get, let's say, for example, COVID, if you are becoming serious, then what happens is that, as I told you, the virus enters through your respiratory system. It is infecting the cells which are lining your lungs. Those lining cells are called as epithelial cells. So they first get into the epithelial cells, and when virus is inside the epithelial cell, our immune system targets those cells and then kills it. So the dead cells are falling into our lungs. So when they are falling into the lungs, it can clog the air sacs. So when the air sacs are clogged, it means that your surface area is no longer available. So which means that enough oxygen is not getting into your blood, right? So when enough oxygen is not getting into your blood, it can cause multiple things. For example, enough oxygen may not go to your brain, so you will become sluggish.
Brain
You will,
requires a whole lot of oxygen.
yeah, multiple organs can fail. I mean, tissues can die. When tissues die, for example, bacteria will attack, stress will come.
Yeah,
So cascade of events will occur, and then
yeah. So
it
what's
will
being
lead us.
said is the new mutant variant actually spreading very fast. Earlier there were enough time for medical professionals to look after a patient, but now it's so rapid that you have to report it instantly when you have some symptoms.
Yeah. Actually, although I would say that the clinical studies and so on, we don't have to make this claim very sure. A lot of my friends who are medical professionals say this
Yeah,
from
I'm,
their
I'm
experience.
actually saying from the quotes of various health ministers and health professionals who are saying
Yeah,
nowadays all these things.
yeah.
Okay, all this, of course, need to be validated properly by a
No, that's what I'm
scientist.
saying, that maybe we will prove to be correct, maybe we will prove to be wrong. But what is coming from the ground, people who are actually treating patients, is that earlier when some patient is directed as positive, it takes many days before that person actually starts craving for oxygen. The oxygen level falls, and then even after the oxygen level falls, it takes some time before you actually have to put that person on ventilator. But now they say that the rate at which, for example, the oxygen incapacitation occurs is very fast. People are to be put in ventilator very fast. That's something that they are saying. Second, they are saying that earlier, young people, people below, let's say 35, very rarely had to be put on ventilator. But they say that today, a large number of them are young people. In fact, this particular observation is not only in India, it's also in UK. With this UK variant, even UK, quite a number of medical professionals are saying that compared to the earlier time, more young people are getting affected. That's something
Yeah.
that is also we are
Yes.
seeing.
Um,
That
uh,
we have to do with the fact that this UK variant is having high infectivity. Basically, it means that more number of copies are produced in the cells.
Much faster.
The virus produces more copies. So, which means that very quickly it can infect more cells. Okay, so that's possibly one reason.
Yeah. Thank you, Mr. Venkateswaran. We are living through a very difficult time,
Yeah,
and we're trying to understand as much as possible. And I hope our viewers will benefit from all of this, what you said. You have really explained what we are facing today. Thank you very much. Bye.
thank you. Thank you.